Hello, this video is about blood typing. Now, in order to understand blood typing, we need to be comfortable with three terms: antigen, antibody, and agglutination. Now, an antigen, which is represented by these blue lines here on the red blood cell, is a large, unique molecule that is used for cell identification. So this is something you talked about back in Bio 23, when you were talking about membrane proteins or glycoproteins. So these are glycoproteins which are unique to that particular individual. And so that's called the antigen. Now, antibodies are proteins that are produced by your B lymphocytes, your B cells, the same antibodies that are produced when you get sick. Now, antibodies, which is what this green structure is here that looks like a Y, antibodies specifically recognize and bind to antigens. So if this were antigen Q and this were antibody Q, then this antibody would bind to that antigen. Now, often we have antibodies, if you can see this green structure here, that have multiple arms sticking out. This one has 5 different antibodies that are joined together to form a pentamer. And when you have that kind of antibody that recognizes an antigen on the red blood cell and you put them together, then that antibody can bind 5 or more red blood cells and create a clump. That clumping action is called agglutination. So when an antibody sees the antigen that it's specific for, it causes agglutination. Now, let's talk about the different blood types. Your blood type is controlled by 2 different genes. One of them determines your ABO group, and the other controls the RH factor. Now, RH is just short for rhesus monkeys, which is where this factor was discovered. And each one of these, the genes that controls these, produces one of several options. In the ABO group, you either get the gene for the A antigen, or the B antigen, or both the A and the B antigen, or none of them, right? And depending on which of these gene profiles you have, we say that you have type A blood, or type B blood, or type AB blood, or type O. So that's pretty straightforward. Whichever antigen you have the gene for, that's the blood type that you have. But you probably are aware that our blood types usually have the word positive or negative after them. And that has to do with the RH group. Now, this, in the RH group, you either get a gene that gives you the D antigen, right? Don't ask me why it's called D. Or you get no D antigen. Okay, slightly simpler than what we saw with the ABO group. However, we don't say that I have type A D blood or type B no D blood. Instead, we say that if you have the D antigen, you have a positive blood type. You are positive for the D antigen. And if you have no D antigen, then you are negative, meaning that you are negative, you tested negative for the antigen. So let's take a look at what some of those blood types would look like. All right, if I have, we'll make our red blood cells red. Wait, that one doesn't work. If I have a red blood cell, and on that red blood cell, I have an A antigen and a D antigen, then that would mean that this blood type is type A because of the A antigen and positive. It's A positive because it has the D antigen. if I have a red blood cell and it has the B antigen and only the B antigen, then we would say that this is type B negative because it is negative for the D antigen. Now, we may also have a red blood cell that has both A and B, and that would be type AB. And because there's no D antigen on this, I would say negative. If this did have a D antigen, then it would become AB positive. I also might have a red blood cell that has absolutely no antigens on it. No A, no B, no D. And this is type O because it doesn't have A or B, and because it doesn't have D, it's O negative. Now, if it did have the D antigen, then we would say it was O positive. So those are the basic blood types. Now, how do we figure out which blood type you have? For that, we have to use antibodies. And for now, we're just going to consider that these antibodies are something that you can purchase from a supply company. But I will talk shortly about how they are made in our bodies and when they are made in our bodies. All right, so the antibodies, that we use for blood testing are anti-A, which recognizes the A antigen, anti-B, which recognizes the B antigen, and anti-D. Now, remember that an antibody, when it encounters its antigen, will cause clumping. So if we mix a blood sample that we don't know the blood type of with each of these antibodies one at a time, and we look to see when it clumps, then we will know that what our blood type is based on which antibody clumped. Whichever antibody clumps, that's the antigen we have. So let's take a test tray. You would be doing this lab in the lab if we were meeting in person. And let's say that there are 3 wells in that tray. And in this one, I'm going to put anti-A, a drop of anti-A. This one, I'm going to put a drop of anti-B. And in this one, I'm going to put a drop of anti-D. And then I'm going to take some blood. So here's our tube of blood. And I'm going to put some blood and mix it in with the antibodies. Now, initially, it should just look like a drop of blood, spilled blood. But if we see within a few moments clumps developing, starts to look chunky and spotted, then that means that an agglutination reaction occurred. So if we saw this, I added anti-A to this one and there was no clumping, then that means that there is no A antigen. When I added anti-D and there was no clumping, that means that there is no D antigen. But when I added anti-B, I saw clumping. So that means that there is the B antigen. Now, if I draw that red blood cell, so I have no A, B, and no D, so it just has the B antigen, that means that this is type B negative blood, all right? There's an exercise in the worksheet and also in the PowerPoint slides that lets you go through each one of these combinations and predict the blood type. or to see the blood type and predict what the reaction would be. So if this isn't making sense to you, I strongly recommend that you go and do that for blood typing. All right.